All right, good morning, and thank you all for being here, and thanks to many of you who we've met over these past few days, and the wonderful children that we've been able to work with since really, I guess, our first child we worked with was Wednesday, so thank you so much. I'm Barb Smith, if I haven't met you, and we're from the University of Florida. And I really have the great fortune, we have to thank Casey Childers for getting to know the wonderful people in this room and their parents. Casey saw Dr. Byrne at a meeting and was able to make some connections and some similarities between our particular long-standing area of expertise, Pompe disease, and his area of interest, X-linked myotubular myopathy. So today, we'd just like to introduce you to ourselves, since this is the first time the big team has been here. And we'd like to draw similarities between our longer-standing area of expertise, Pompe disease, and what we sort of call its cousin, X-linked myotubular myopathy. We want to talk a little bit about the differences between observational studies and clinical trials, which is what a lot of people in this room are working very hard to get to. We want to share our path to clinical trial for a gene therapy study to the diaphragm in Pompe disease. I'll give you just a quick summarization of how that's looking so far. Discuss our current and future thoughts for clinical trial outcomes, and share the lessons we've learned so far. So just to introduce you to the University of Florida, please think beyond sports. We do a lot of other good stuff there, okay? We do have a center of excellence, a clinical excellence. Florida hosts one of the about 200 MDA interdisciplinary clinics in the US. We are also a top-tier research institution. 2011-2012, the university was the recipient of about $664 million in research funding, and we're one of 61 institutions awarded a Clinical Translational Science Award consortium by the NIH. Also, we have the UF Powell Gene Therapy Center, and it really is a pioneer in gene therapy. Dr. Burns, not Dr. Byrne, but Dr. Burns, is who we can credit the discovery of the life cycle of AAV to. Also, Florida was the first use of the AAV vector in animals and in humans. And then Dr. Byrne, who's here in this room, who I will introduce you to shortly, was a pioneer of the first use of the AAV vector in muscle and heart disease, and also the PI of the first human clinical trial of the AAV gene therapy vector to the diaphragm, and that's our study we'll talk about today. One more thing I think you need to know about UF is that we are really a leader in bringing research into commercial success. So research doesn't really help that many people if we don't get it out of the ivory tower. And so this institution has a very strong track record of that. 2011, we were 10th in number of patents brought in. We were 8th in number of startups in the US. And also the Sid Martin UF Biotech Incubator was just rated this year as the #1 university founded biotech incubator in the world. So we're kind of proud of that as well. But we want to step back and talk a little bit more about gene therapy and what we've been working on with our cousin disease, Pompe disease. And for that, I'd like Dr. Byrne, since he's really been our leader in this area.
Thanks for the introduction, Barb, and thank you all for including us in your MTM1 family. And as Barb said, thanks to Casey for making this connection, because we do see a lot of similarities in the problems that children face in this condition, which is an inherited muscle disease as well, but due to another gene defect that leads to glycogen accumulation in muscle. And as a storage disease, it has some things that are quite different than MTM1, but particularly the cardiac effects are quite severe in the youngest patients and one of the principal causes of early mortality in the patient population. But many of the final common pathways that lead to ventilatory failure and the ways that we can measure that and expect changes with specific therapy are quite similar. So, if I can advance here, I'll do it here, yeah. Excuse me. So, the relationship between the gene defect and the degree of loss of function is, again, very similar in all the genetic causes of muscle disease. As I mentioned, the heart defect causes increased cardiac mass and heart failure. The musculoskeletal problems are really very profound weakness with early loss of motor milestones and even observed in infancy. And then the respiratory problems that are very characteristic also of myotubular myopathy. So we can draw some common characteristics that, as Barb mentioned, may be a related between these two problems. First of all, they're both rare conditions. Early limb and respiratory weakness are the features of both conditions. A single gene defect is the cause, and approved therapies really have not been available that allow for significant reversal of the symptoms that are most problematic for patients. And these target genes, both Myotubular and GAA and Pompe disease, are small enough to fit within the AAV vectors that what first motivated us to look at AAV as a tool for correcting genetic muscle diseases. And therefore, this makes really an ideal opportunity to use the lessons learned from one disease to benefit the other. And this really goes back quite a number of years. As Barb mentioned, Ken Burns, who was the chair of microbiology at our university, really discovered the life cycle of AAV, and several of his students and colleagues facilitated the discovery of this as use as a vector. And that led me to move from Baltimore to Florida in the late 90s to work with that group and help bring those basic science discoveries to the clinic. And that's really been the focus of our center since the early 2000s. So we've actually done a number of clinical trials with this vector system, 7 to date actually, where the vector manufacturing has actually been done at the university. But also, as Barb said, they're really to really make this sustainable for the patient community, we want to have a means to which we work with our commercial partners to make this really durable. And you know, the preclinical evidence, as you've heard from Casey, as we've developed in our own Pompe example, is really very strong. And then the goal, what I give you a preview of, and Barb will provide the details, is how the clinical trials actually can lead to real significant benefits for the patients. And to kind of outline how you go through that process, I think it's important to highlight, because you're really at the early stage in this community of moving from those observational studies that define natural history to real controlled clinical trials in which the outcomes are carefully monitored, that provide evidence to the regulatory agencies that the effects are really due to the treatment, and therefore those well-controlled studies lead to approval of the product, which lets it be available, makes it available to the wider community. So in observational studies, they're just no intervention, but really natural history, as has been described. You may get more participants, but not as much specific data, whereas the type of data that's collected in a clinical trial really has to be so carefully selected that it's verified as being reproducible and helps establish a pathway. So the data collection may be changed during the course of a natural history study, but that's not the way one would do a clinical trial, in which you have to define upfront what's being collected and carefully follow that plan. This observational study certainly is critical in rare disease studies where treatments that are not given during the clinical trial may not be ethical. If it's really felt that the treatment has a significant impact on the disease, you wouldn't want to withhold treatment from any of the patients participating. But I think there are now a good federal, at least in the US and certainly in the EU, structure for getting approval based on small disease studies where initially safety and efficacy is done, established, but earlier phase trials in which we can also possibly seek approval of the product. And the conclusions from these observational studies are not always so concrete, but the conclusions that we need from clinical trials is that there really is a direct therapeutic benefit of the trial. And this just gives you an idea of a review of what we went through in the Pompe story to initially generate a mouse model in 1998 with collaborators at the NIH, and then show a series of preclinical proof of concept studies between the early 2000s and then the current clinical candidate vector that we have was first described in 2006. And you can see this is all just the progress, the rate of progress that comes from NIH grants. When we mix in sponsorship from an industry collaborator, this timeline hopefully can be compressed, and I think that's what you're about to see take place going forward in MTM, because we really didn't have commercial sponsorship of any of these activities. So ultimately from 98 to 2001 when the clinical trial started. And so this is what this trial was called. It was an early phase study because that was the regulatory environment. There is an approved enzyme replacement therapy for Pompe for which the first industry-sponsored study was in 2002. So really, if you put that in the backdrop of this, you see that there's again another similarity between the management approach. So we sponsored this study ourselves at the university with all NIH funding, a mixture of funding sources because really one type of grant couldn't do all of this. And we first got approval from one of the regulatory agencies in 2008 and FDA approval in 2010. And we've actually just completed the first phase of this study in between a brief introduction and that I'm going to give, and then Barb will go through some specifics. So for this study, the question we wanted to ask is, could the gene being delivered to the diaphragm, is it safe, and could it improve ventilatory function based on what we knew about what causes weakness in these patients? So the sixth patient is not listed in here because it was only dosed 2 weeks ago. So this completes enrollment of the first phase of this study, and we have received approval to do another 5 patients to round that out. And you can see the age at diagnosis, and then the age of mechanical ventilation in months shown here, and when we enrolled the patient. So a variety of levels of function, and the study design is fairly simple. For one thing, I think it's important to emphasize, I think Barb will make this point in her part of the presentation, patients with prolonged dysfunction, muscle function problems over many years are not going to simply switch from their severe disability to normal function in a matter of days. And so we wanted to also test what is the rehabilitative potential of some interventions that are commonly used. So we had a prehab period of looking at particularly respiratory strength training between minus 60 days in the day of gene dosing. I won't say a lot about that other than that it's important to understand the baseline function of the optimal standard of care prior to dosing, so we can attribute any of the changes to the gene transfer itself and not to this care given during the trial. And then a series of efficacy studies about respiratory strength and endurance and off-ventilator tolerance, and Barb's going to talk about this part now. And so I can summarize briefly the safety outcomes is in that this vector, now delivered to over 300 patients for a variety of diseases, has been extraordinarily safe. So we see no safety signals from these early phase studies, and in fact it helps us make the argument now that there's been sufficient cumulative experience to move beyond safety studies where information gained from one study can be used to support a higher dose or more efficacious dose in another patient population. And that's, I think, another thing we can share between the two conditions and hopefully move the timeline along a little faster for MTM-1. So I'll give it back to Barb, and she can talk about the efficacy outcomes of these trials. And thank you all for having us.
Okay, so let's talk a little bit about breathing, all right? So respiratory function was our secondary outcome for this clinical trial. That's the efficacy component of the clinical trial here. And Dr. Byrne mentioned that if we put me on a ventilator for 12 years, as one of our children who was enrolled in this study if we put me on a ventilator for 12 years, and I didn't have to use my diaphragm, I probably wouldn't be able to use it very well. So there's not a whole lot known. We've got some information known from the intensive care unit and the ICU-related research about being on the ventilator for short periods of time, days or even weeks, and yes, that diaphragm, it atrophies very, very quickly, even without a neuromuscular disease. And we just don't know about what happens to the diaphragm in the time range of years. So we wanted to look a little bit before gene dosing, and that's where we provided the conditioning exercises. One was to make sure our kids' breathing function and their diaphragm function was as good as they were going to get without the gene therapy. And the other thing, we sort of wanted to prime the pump. We wanted to prime that muscle, get it ready. The second phase was the gene therapy after dosing, and they continued to exercise for this full year. So kind of leading into that concept that clinical trials, we ask a lot of our patients and their families. So looking at efficacy endpoint, the question we really wanted to know beyond safety is, were we able to see any changes in breathing? And we derived our outcome measures from the ICU. I think a few of you had the chance to meet Dr. Danny Martin from our team on Friday. I think he worked with a couple of families that were in here. And Danny is an expert in measuring function and training patients both from 1 month to 80 years, really, in the ICU. And so we selected measures that were validated in the ICU to look at these three domains of function. All right, we can also simulate some of these measures in preclinical models. We'll show you that later. And we've used this, Danny and I have used these in other neuromuscular diseases, namely Myotubular and myopathy and Duchenne. So when we looked at our kids, they came to us for enrollment and we studied their baseline function. We saw that their function was quite restricted. So these are all of our 6, we've got these 2 folks ended up not getting dosed. They had to be disenrolled for other reasons before dosing. But our kiddos who came to us and worked with us at initial enrollment all had very low tidal volumes with their best effort off the ventilator. These dashed lines you see here would be resting breathing that you and I would generate. This is normalized to our body weight. So we had our kids off the vent. I'm going to lower the microphone. We had them. You don't want to hear me heavy breathing into the microphone, I promise. But we had them generate just the biggest, fastest, toughest breaths that they could on a maximal voluntary ventilation maneuver, and really kind of have them rev their engine, rev their breathing engine. And we can really ramp up our breathing 20-fold at least. These kids, at their very, very best, were unable to reach resting breathing volumes. We did not do this particular test in most of our boys this weekend. One test we did do with our kids this weekend was the maximal inspiratory pressure test. And that was that manual muscle test for the inspiratory muscles. So we had them pull in against that closed valve and try to make a lot of tension with their inspiratory muscles. And again, so we can see these are sort of our upper and lower limits for what we would expect this group of 6 to be able to do, and they didn't quite make it that far. So being on the ventilator for 1 to 12 years and having a neuromuscular disease resulted in their inability just to get quite to those levels. So wanted to show you, if you're squeamish, this is just our dosing strategy. Thought some of you might be interested. Look away if you're not. But we'll get you started here. So you are inside of the left side of the thorax, and you're looking down on the diaphragm. And that's what we're seeing here. Toward the top of the screen, you're going to see a needle coming in through the costal space there. There's Dr. Islam, and he's going to find the area of the diaphragm where he's going to place that needle right in the middle of the diaphragm muscle. Right? The AAV vector with the gene inside of it is at the other end of his needle. He's going to make sure he's well inside the muscle. You can see this little blistered area here. There was one area that was already dosed at the front of the diaphragm. This is the middle, and you'll start to see this blanching. Nice slow push. You want to get all that good gene therapy right in that middle costal diaphragm. All right. And then once all of that's in there, you're going to see that needle slowly retract. And we would, then after Dr. Islam was finished, we did a third dose, all right, toward the back of the costal diaphragm. So we dosed each of these children 3 spots on the left, 3 spots on the right. And there you have it. All right, so a year later, what did we see? Did our kids' muscle function change after gene delivery? So since, as you saw, every kid's bar was a different height, right? So we normalized their performance to themselves. And that's where our 100% comes in here. So the day before, well, 2 days before, they had the pleasure of working with me and going through all those wonderful tests that your kids did. And so that was their baseline function. And at these 90-day time points for that first year after, we normalized their performance to their baseline function. And we see some of our kiddos, based upon how they were feeling, if they'd been recovering from illness or had a bad day. Some of our kiddos, like this little guy here, had a little bit more up and down. Some of our kids are a little bit more up. But we did see those tidal volumes decrease. The mean was about 50% increase off the ventilator. Wrong way. All right. Off ventilator endurance, time and volume, that consistently everybody got better. This kiddo here is about in the middle between the green and the yellow line here, finishing out to the end. So our kids increased anywhere from about 200% up to 3,000% of their baseline. So this kiddo here is went from about 8 minutes that he could tolerate being off the ventilator to about 3 hours at the 1 year point. So we had a different rate of response, but everybody did make some changes. And some of those had some pretty meaningful benefits for our family. I'm not going to give you every kid's story, but I'll give you 2. So kiddo 103, his family, he's of the age where with little boys where baths are the most horrible thing on earth, and just cries and hates them, and just we don't want to do that. So a slippery boy in a tiny skinny little bathroom, it can be a difficult thing with a 3-foot tether on a ventilator. So they incorporated their off ventilator time, since he was crying and mad anyway and breathing heavy, into his bath time. So he would come off the vent, he'd get undressed, he'd get in the tub, he'd get cleaned up, he'd get back in bed, he'd get dressed, and then he'd get the ventilator on. So that was actually really helpful for mom, not to have to slip over a ventilator while she's bathing her child. This child, 101, this was the child who, between day 90 and day 180, we got a call from the parents, and they said, hey, we went to the park again today. They have a park near their house, and we found out that they went to the park for the first time without their ventilator. They felt that their child was stable enough and strong enough that they could leave the home and keep the child off the ventilator. So that was a really big step for them as well. So our conclusions, we saw just at baseline coming in that we had some residual function in the diaphragm, in the respiratory muscles, but it was quite limited compared to unaffected children their age. we really didn't see, I didn't share this part of it, but we didn't see a clear benefit with any of the dosing exercise, the pre-dosing exercises. So exercise alone wasn't going to change their function a bit. The FDA is quite pleased with our safety and our toxicity studies. We're glad for that. And we're encouraged by our early findings, but we cautiously optimistic. So we know, as I mentioned before, that just being on the ventilator a long time can cause a lot of weakness and dysfunction. But also, this was what was classified as a local gene delivery. One muscle. Now the diaphragm is a very, very special, important muscle. It's my favorite muscle. But, breathing is really a complex motor program, all right? So even when we're resting, breathing, we use other muscles. We use some of the muscles up here at the top of our rib cage. We use some of these scaling muscles in our neck, just with resting tidal breathing. So we might need a little bit more of a sophisticated dosing strategy, more of a regional strategy, or maybe even a systemic strategy to better target the entire pump. Okay, so I want to shift gears just a little bit to clinical trial readiness, because that's something that we've worked through and you guys are working through now, and we'd like to help some with that because we know a little something about measuring breathing at the University of Florida. So we would like to study our respiratory outcome measures that we're using in Pompe and move those forward into MTM-CNM. And really, are the tests feasible? Can they detect a change in function? But even stepping back further beyond that, what is the expected function? So if you do a literature search, a PubMed search, and you're looking for control of breathing and respiratory muscle function in MTM-CNM, you're not going to really find any hits, okay? So we're blazing a trail here. So I want to focus on a couple of measures, all right? Maximal inspiratory pressure. All of our kiddos who came to see us underwent the maximal inspiratory pressure test. So I kind of pulled that one out. And it's very well validated, both in kids, adults, ICU, out of the ICU, as a very valid estimate of respiratory muscle strength. So those of you who have participated, remember, our kids could breathe out a little bit, but when they pulled in against that valve and they made a lot of tension. And, yes?
I'm sure you did MIPS on the other in the study.
Yes.
I would think that would be the primary but anyway,
what's the question?
The MIPS, we had our most modest change. Oh, I apologize. So Casey asked, MIP is probably an important thing. MIP is maximal inspiratory pressure. And did we see the similar slope of change in our Pompe clinical trial? And we saw some change, but it was our most modest change. So, and I think this is why, Casey. So, the maximal inspiratory pressure, your diaphragm is recruited, but a lot of what else is recruited. So, the diaphragm pleads for help on this maneuver. It's the most you can make pulling in, and so it's a very generalized measure. So we use a lot of extra diaphragmatic muscles, like our intercostal muscles. So if we had perhaps, we just didn't want to do this, we can drop pressure catheters down and get an esophageal and a gastric and do a transdiaphragmatic pressure, and I think that would have been more informative. But this was a global measure. It's very important, though, I think, especially when we're just sort of blazing the trail in MTM-CNM. I think this is going to be a really important measure for us. But that's an excellent question. And if we're looking at regional dosing, I think this is going to be quite interesting. So, so you can see we generate some of these big peaks when kids pull in hard against that valve. And our highest, our most negative pressure that we make, inspiratory pressure is negative, that's our MIP, okay? So I confess, I stayed up a little late last night. I wanted to back up all the great data we collected last night, and then I was looking at it and said, wow, wouldn't it be great if I could give some data this morning? And so that's what I did. So this is just a summary. This isn't the prettiest slide, but this is a summary of the 5 kids who completed clinical trial in Pompe. And this is 12 of the kids who have a similar ventilatory function with MTM-CNM. So we tested a couple of folks who were not ventilator dependent, didn't include them in this particular score. This deep dark line in the middle, that's the middle value, okay? And so our average for our gene therapy patients in Pompe was about 8, was about -8. And the average of the boys we tested yesterday was about 14. So actually you guys did a little bit better than our gene therapy patients. And then this is and these are sort of the upper and lower levels. And so these are our unaffected kids, control sample. This was at inclusion, yes, so this was before gene therapy. And so if we just look at those individually, I really wanted nice individual points so everybody could look for their respective warrior. But this is Pompe, so we had, as you saw earlier, a lot of variability. We see a little variability also in MTM-CNM, but that's the same scale, so we see that we get a lot more blue on this particular graph. The other thing that we've been measuring is respiratory kinematics. And the question we have for that is, which muscles are better preserved in MTM-CNM? So is the diaphragm the muscle that we really have to target? Or maybe some of those extra diaphragmatics or abdominal muscles, are those more or less preserved than the diaphragm? And then also, how when our kids sense that they need to do more of the work and they begin doing more of the work, how do we see that their patterns of recruitment change as demand increases? I'm just going to show you a little bit. We got permission from our cover model up here wearing our kinematic belts, okay? And this guy here is a special warrior because we showed up at Will's house a year ago, and he was gracious enough to really be the true trailblazer of the room. First kid we've tested, and he was just phenomenal. He blew Dr. Byrne and I away. We were so proud and amazed with him. So thank you, Will. Thank you, thank you, thank you, thank you, Will. So you can see there's a belt around Will's chest and then one around his belly. And so when he asks for air, all right, and he pulls in air and his diaphragm descends, we would expect his belly to move out a little bit, all right, and also his chest to move out a little bit. So we can see relative rates of diaphragm descent and how that changes, and even the timing. I'm going to show you just an example of what this looks like in one of our kids in the Pompe gene therapy study. So our kids also get EKG. We just watch rate and rhythm as that happens. And this is the child on full ventilator support. This is the chest band. This is the abdomen band. And then this is a total that we can correlate with volume, with inspiratory volume. And this is sort of lower and upper, end expiratory, end inspiratory. Same thing here, end inspiratory, end expiratory, inspiratory, inspiratory, expiratory. So what that says is the displacement when we fill our lungs with air, and then when we relax, and where we start from. So this child, the ventilator was doing most of the work for him. He had this nice, slow pattern, big volumes. And then when we took the settings down and he asked for more air, we see a lot smaller volume, okay, generating a lot smaller tidal volumes. And we also see a lot lower displacement, all right, abdomen and chest, but both on the end expiratory side and the end inspiratory side. Shallow, rapid breathing pattern. So we've conducted that again in 12 of our kiddos this weekend. We're so thankful and happy that I didn't have a chance. I wouldn't even have one child done if I had stayed up all night to give you that. So I didn't. But we have some lessons that I think that we've learned between this weekend and the work that we've done so far in the Pompe world. We really do need to think about clinical trial outcomes now. You got to pick them well before you go to the FDA and ask for their permission. And as I think you're getting a sense of from this full 2 days, really the clinical trial is just the end product. There's this whole mountain, of the whole ocean of research that the clinical trial boat is floating on, right? So we are so thankful to all of the people who are in that ocean with us. It does take longer than we think it will. Hopefully with our experience, some of the experience we've had in Pompe and some of the kind of the shifting sentiment a little bit of the FDA, it won't take as long as it did for Pompe, but sometimes things do, delays and roadblocks come up, and we just want to be prepared for that and not have a specific time in your mind when we're coming to you. Also from the patient family side, our clinical coordinator really wanted me to emphasize this, that it does, we totally get that it requires an enormous commitment from our patients and their families. They need to travel to come for gene therapy, and they need to travel to come for at least one study endpoint. But for instance, our Pompe subjects, they conducted training 3 times a week with me for a year. So that's a time commitment, big time commitment. And also the testing. So there are certain study blood draws, study imaging tests, some study functional tests that we just needed to get. Those are crucial to obtaining approval, right? And then also participation in a clinical trial really involves long-term. It's a long-term relationship with your study institution. So our patients, Dr. Byrne, is it 20 years we have to make reports on them? So you'll be hearing from us if you ever are in a primary clinical trial. And then also just keep this in mind. Of course, it's great to have social media and to be able to network and to touch base. But sometimes, like, not everything you read is true, right? Sometimes rumors can overtake where things are coming from. So if you have a question, you hear something, oh, they're doing this, this study's open and I haven't heard anything about it. Call up, University of Washington or call up University of Michigan and ask, okay? That's going to be your best source. So I'm so glad that you guys are able to keep the communication lines alive, but if you have questions, please feel free. We're happy to address them. And then finally, we just want to acknowledge we've got we probably missed a zillion people on here. We've got a big clinical team. We've got a massive bench team that's helped really move forward to this point. We're very, very grateful to the funding sources who have supported the Pompe clinical trial and continue to support us as we move forward. And we're available for questions.